Thank you very much, Daryl. So the issues that surround research in this area are, of course, very complicated. We have a traditional model that we use to address any research question, and at the beginning it's very similar to what we do in complex interventions for quality and safety. You have to identify what the issues at stake are. You have to target either a disease or a syndrome. You have to define it for operative purposes. Many of these syndromes are really undefinable. The deteriorating patient has all sorts of fuzzy edges on both sides, but you have to define it in some way. Then you define the syndrome, you define the epidemiology, and once you know the epidemiology, then you understand what the outcomes are that might be associated with the condition you want to study. And once you know that, you think, okay, what are some logical interventions or technology or devices or medical treatments that we can use to help diagnose it and treat it. And then you are ready, if you wish, to undertake interventional research. And that's where things get a little bit more complicated. So the first bit is the same, no matter what you're dealing with, whether it's complex interventions or whether it's just a new drug that you want to develop or give for a particular condition. But then, after that, things get a little bit tricky, because in the traditional model, then if you want to study a drug, then you move into first doing feasibility studies, then doing some pilot studies to show that you can deliver the drug and that everything is okay. And you do phase 2, small randomized controlled trials, maybe single center or 2 or 3 centers, to show that you can deliver the intervention is safe, and there is an inkling, some kind of surrogate measure that the intervention is going to be helpful. And then if you find some evidence that the intervention might be helpful, you might be able to move to phase 2B studies that give you a little bit more of a sense of an effect. And then you might be able to move to phase 3 studies to definitively answer the question. However, there are tricks and traps. Life is not that simple. Even in the traditional model, you'll have all sorts of different views. There'll be disagreements about the target, disagreement about the definition of the target. Often the epidemiologic data is not that robust. It's often just a single center or a particular region. There is often limited information about the outcomes. There's uncertainty about what outcomes ought to be relevant to the questions you want to ask. There are controversies about what interventions are likely to succeed and what interventions are likely to fail. And of course, there is the usual smattering of belief, dogma, politics, sociology, control agendas, power, and money that besets all human activity on this planet. In this world, there's been the arrival of the evidence-based paradigm, and that has been in response to the fact that too many things in medicine and surgery were being done and are still being done without suitable evidence, even though they've got quite significant implications for the well-being of humans. And so the evidence-based paradigm promoted the view that we need better evidence, not just let doctors decide. We need to have objective, robust, verifiable, reproducible, high quality, statistically endorsed and supported evidence. It's not enough to just ask experts. They are wrong a lot of the time. And they created a paradigm. That's a paradigm that is open to challenge, like every construct by human beings, but nonetheless, it went through a series of recommendations that were targeting particularly the elimination of bias, selection bias, ascertainment bias, confounders. And that was the major driver for evidence-based medicine. And there's a consequence of the desire to avoid bias. A whole structure was created, which is the pyramid of evidence hierarchy. So you have the large multi-center international double-blind randomized controlled trial. Sometimes you cannot blind the intervention. That's okay. Then you have studies that have got a moderate chance of type 1 and type 2 error because they're relatively small. And a type 1 error is when you find something but it's actually not true. And type 2 is when we don't find it, but that's because you don't have enough power to see it. And then you start having randomized controlled trials where placebo cannot be applied for a variety of reasons. And then you have contemporaneous interventions that are not randomized. And then you have the before and after clinical trial. And all of these approaches to trying to find, quote, the truth have got, if you wish, a different level of bias, a different level of robustness, a different level of certainty, and they're classified in different ways. And so, given all of this stuff that's going on, how in the hell are we going to know when something is true or not? Well, we can't. We don't. We never will. We can only approximate the truth by defining it in an operative, statistical way. It's a bit disappointing, but the truth, the cosmic truth, is not available to human beings. So what do? We define the truth statistically, and I'm sure all of you are comfortable with the p-value, but of course all those that do research think, really, come on, give us a break. So if the p is 0.049, it's significant. If it's 0.051, it isn't, really? Come on. That's clearly stupid. But nonetheless, it is often rigorously and religiously applied because it is a convenient and simplified way of understanding the world. And then people have introduced the confidence intervals. Well, it's the same thing with a different name. If the confidence interval crosses the boundary of 0 or 100%, then it's real, and then it's not real. It's just another game for defining what should be acceptable. People have said, well, we should do the Bayesian approach. We should say, oh, well, the pre-test probability is really important to believe that what you find is real. But then no one can agree what the pre-test probability is for any specific intervention you might even think of. So that's gone down the window because it doesn't really work. And of course, there is the issue of power, whether you have the ability to see that which you're trying to see. What's the chance that you will see it if it's there? And it's really, again, complicated because power is money. So if you want to do a trial that's got an 80% power of being able to see a specific outcome, then you're going to spend X amount of money. If you're going to go to 90%, it's going to cost you an extra $1 million. You can see that it's complicated. And then there are lots of things that trials never tell you about. Biological plausibility. I mean, we can do a large multi-center randomized controlled trial of having curtains that are blue versus red in the ICU. And we might even be able to find that it shows a statistically significant difference in mortality. But none of you would believe it, right? Because it has no biological plausibility. It doesn't make sense. There would have been some bias. People knew that the blue ones were coming from the company they liked, and they kind of twisted the outcomes in some way or another, consciously or unconsciously. There are issues of external validity. If it happens here, is it really going to happen somewhere else? Applicability, can you actually do it? And all of these issues that are gigantic, each one of them requires an hour of discussion, in the conventional environment become incredibly complex when you have complex interventions. So what do? Complex interventions often are treated by observational approaches. We obtain data at baseline, we make the change, we obtain the data again, and Daryl was showing you very lovely segmental analysis data, and then we see what happens after we've introduced this change. This is what we call a sequential before and after trial. It's not blinded, it's not randomized, and of course it's got a huge Hawthorne effect. Hawthorne effect is when people's desire to make something happen is what really makes it happen. Right? But hell, this is exactly what complex interventions are about. They're all Hawthorne stuff. And so when people criticize it because it's a Hawthorne effect, you would say, duh, that was the whole thing. We wanted a Hawthorne effect. This is a case in discussion, a central lines, paper in New England Journal of Medicine, intervention before and after, a series of steps to make sure you don't do spit and polish when you put the central line in, that you apply chlorhexidine and you gown up and you're careful, et cetera, et cetera, et cetera. And it's been criticized because it didn't have all sorts of aspects that might be confounders. But the reality is that an intervention was applied, a difference took place, things were better. Why is that a problem? Why would you not want to do the same? And so when you come to the medical emergency team environment, it all becomes really tricky because there are interactions between the team, the home team, the ward nurses, and then if that wasn't enough, there are interactions between the patient and all of these teams involved. And if that wasn't enough, then the families are part of all of this, the way they interact with the staff and the ward and the patient and so on. So you have a very complex series of potential confounders, and it doesn't lend itself to what would be the traditional drug trial. And you can't go out there and just put this thing in the wards and say, okay, guys, you've seen those things? Yeah, just go do it, right? Because it doesn't happen. Because, again, the complexity of triggering and responding to a patient requires identification. The calling criteria are a methodology to help you. They're just a reminder. They're just a tiny little bit of the medical emergency team complex. There's activation. There is team involvement, other teams, critical care beds, home team. It's just massive. And you cannot try and address it with a randomized individual control trial. And as you know, we've used before and after interventions, and they've all shown an effect. But then if that's what you do in a single hospital, people will come back to you and will say, well, okay, that's what happened at the Austin. There are a bunch of do-gooders, and Daryl and Rinaldo spend all their time to try to save lives and getting all upset if people don't get treated properly. But can you take us somewhere else? Can you take us somewhere else? Or maybe the Austin is so bad that even doing a little bit of medical emergency team activity makes it better. No wonder they're so bad to start with. In our hospital, that wouldn't possibly happen. And so you have real problems. And then to try and address these problems, that's how the MERIT trial was developed. And that was a way of trying to go beyond the single hospital, like Liverpool Hospital for Ken Hillman, or the Austin for us, and to try and understand whether this would work somewhere else. But if you do that, and you try to do randomization, you can't randomize patients because it's logistically, technically, statistically impossible. So then you randomize the hospitals. But if you randomize the hospitals, you get the cluster curse. And the cluster curse is that each hospital is an individual. And then you lose power. And then all you have to have is a lot of variability in the hospitals as part of their natural fluctuations in cardiac arrest and mortality. Then you lose power even more. And then if you have some hospitals like a small country hospital participating in a trial like this, where the events are small because they don't have that many patients, you lose power again. So you end up with the MERIT trial that had a 25% power of seeing a 30% decrease in the outcome of interest. So you're spectacularly underpowered. And then people that don't want to apply this kind of technology to make people better will fight against you and try to understand and tell you that, okay, this thing doesn't work. But then you can go back and try to understand it a little bit more, and you can look at whether there are relationships between the dose, the way you intervene, how often you intervene, and you start seeing things like this. Then in every hospital that was in the trial, the activity that was not associated with cardiac arrest but reviewed the patient was the dose of the preemptive intervention. And the more preemptive intervention you had, the more you achieved a better outcome. And then if you wanted to be cynical, you say, well, that's not good enough. I really want the highest level of evidence before I implement any quality and safety activity. Really? Where is the randomized controlled data about ambulance services? Why are we sending ambulances to people's homes? Where are the randomized controlled trial of intensive care units? Why don't we randomize people to stay in the ward and to go to ICU? They're clearly stupid. And there's a whole lot of stupid things. Where is the evidence that BLS and ACLS and ATLS makes any difference to anything? We will never have it. And let's get real here, okay? We can never prove this stuff. Relax. It's okay. Let it go. Let it go. Frozen. Let it go. We need to accept that these changes are all about leadership, plausibility, common sense, trying to do that thing that we want to do. System changes studies will never tell you what will happen. They never will. They can tell you what can happen if you apply yourself to it. And in the end, you will always need to make a judgment, whether it's about washing hands or medical emergency teams or outreach systems of liaison nursing or line bundles or delirium bundles. You have to make up your mind. That's what you paid for. So this is another example. Washing hands. This is the Austin Hospital. A bunch of do-gooders, by the way. This is the debug thing. Okay, so the infectious diseases were there. They wanted to facilitate hand washing. They look at the MRSA isolate. You can see how it changes. They looked at the episodes of MRSA bacteremia. They see how they change. They look at the Klebsiella and the gram negatives. They see how they change it. This is not a randomized controlled trial. There you go. This is not high-level evidence. They didn't randomize the patients. How do we really know this happened anyway? The thing is, who cares? Who cares? Do you really believe that washing hands had nothing to do with these outcomes? Of course you don't. Of course you don't. Should we reject hand washing? Really? I kind of like spit and polish, but really, come on. Another important thing is registries. I think they're important in the assessment of these complex interventions because registries help us understand the activity, the epidemiology, the outcomes, the ongoing process of healthcare. And it would be really good to have a registry that encompasses, at least to begin with, Victoria. We have a Met call, as best as we can ascertain, every 10 minutes. And we have Met calls, as best as we can ascertain, have got a mortality above 20%. We do not have a registry of this in our state, let alone our country. In intensive care, we have a national registry of intensive care admissions. The mortality of intensive care admissions across the country is less than 12%. There are more Met calls in Australia than ICU admissions. This dissociation is a disconnect about the quality of data, and it is important that it should be addressed. So RCTs are important, they're useful, they're not the only way to try and understand things. System changes can't be blinded. Observational studies are helpful. Cluster RCTs are extraordinarily difficult and typically underpowered and logistically extraordinarily expensive and problematic. And you will never satisfy people that want to believe. Before and after studies are useful. They have a biological and a clinical rationale. They provide evidence of safety. They provide an understanding of the cost, but also they also show the cost of inaction, of doing nothing about problems that beset the healthcare system. Many people will continue to be obsessed with high-level traditional evidence in the assessment of complex interventions. We can't stop them. There are about 100 or 150 people are part of the Flat Earth Society. We can't stop them. It's a mostly free world. People can believe in pink fairies if they want to, but the common sense test should be applied to all of these interventions. And system studies will continue to remain different, will continue to remain absolutely different. Before and after studies will often be conducted to try and illuminate and help us understand what happens or what can happen if we apply specific intervention. We absolutely now need registries because if we're going to apply interventions on a state level or a national level and we're going to do before and after studies, we absolutely need that baseline information to see if these interventions modify the patterns and the outcomes that we're interested in. In the end, I can assure you, most people believe the world is round. In the end, most people will believe that using a bicycle instead of walking is faster. In the end, common sense prevails. But it would be nice if it took a little bit of a shorter time to prevail. Thank you very much.